So we're starting with the histology of the ovaries in the female reproductive system, and we'll be doing all of this in terms of points. The first point is out of the surface epithelium. The surface epithelium of the ovaries is made up of simple cuboidal epithelium, right? Simple cuboidal. Simple over here means that it is single layered. Cuboidal is the shape. If it was stratified, it would be multiple layered, but simple means single layered. And how do you represent a simple cuboidal epithelium? You represent it like this. The height and the width, or the length and the width, whatever dimension you want to refer to, are the same. And you have the nuclei in the center, and you draw the ovary as being almond shaped. Second point is that the ovary is divided into a medulla, which is the inner portion, and the cortex, and this is a way of naming that is true for most organs, for example the kidney. Let's talk about the cortex first, and then we'll talk about the medulla, because the cortex is the outer part. Right, now we can say the cortex is the broader portion, the medulla is the inner portion, it is a smaller area comparatively. So that is the third point, that the cortex is broader compared to the medulla. The fourth point is the division. We divide it into a stroma. Now the stromal portion is, or the stroma is basically compact, and it is richly cellular. There is a lot of cells there. And the second portion is made up of what we know as the ovarian follicles, and they are the spherical bodies you will see in any section of the ovary. And these follicles are irregularly scattered in the stroma, which is a richly cellular and compact layer. Now we'll talk about the stroma now in the fifth point. The stroma of the ovarian cortex is composed chiefly of stromal cells, no surprises there. What else would it be composed of? Stromal cells, because they're making up the stroma, which has been described as being richly cellular, and the cell shape is spindly or spindle shaped. Now collagen fibers, which are the collagen fibers portion, the reticular fibers made up of collagen type 3. Then we have the cells, we have fibroblast cells, which are the most common cells in the connective tissue anywhere. And then we have smooth muscle cells, and they're macrophages, and they're present in between the stromal cells. So collagen, reticular fibers, fibroblast, macrophages. Macrophages are cells of the immune system, and they're the non-specific sort, and they tend to be present a lot in connective tissue, right? And also we have the fibroblast and the macrophages and the smooth muscle cells. Right, we'll be talking about all of this in a while. So this is a constituent of the stroma, and there are a lot of blood vessels too. And you tend to represent the blood vessels. Actually, you use pink for it. So you represent the blood vessels in the stroma. You draw the circle to show it's a blood vessel, and depending on the thickness, it's a capillary or an artery or a vein. And for a vein, you draw it like thinner wall, and the lumen is bigger, but this is an artery. And because this is an artery, so it's, you kind of do it like this to represent the nuclei. Right, so they're also present. Now, talking about the stromal cells in more detail, the fusiform stromal cells of the ovarian cortex, they are arranged in a haphazard manner or an ill-defined manner. These cells have the capability to transform into endocrine cells and give rise to a sheath around each of the growing ovarian follicles, and this sheath is called the theca folliculi. Right? So we know that there's in the most mature follicles there is something known as a theca folliculi, which is a connective tissue capsule having an internal portion and an external portion. The internal portion is the vascular portion, the external portion is a simple connective tissue capsule. And you know that eventually this theca layer is formed by the cells that are already in the connective tissue cells that are already in the ovaries. So now that particular mystery has been solved, because it is the stromal cells, right, which are the spindle shaped cells in the stroma in which the ovarian follicles are embedded. They are going to differentiate. They themselves are arranged in a haphazard manner and then in the cortex, and they are going to differentiate, and they're going to give rise to a sheath around the follicles, the growing ovarian follicles, which is known as the theca folliculi. Right? Now the cells of the theca folliculi, specifically of the internal portion of the theca folliculi, they will actively secrete the female sex hormones, theca interna cells. So that is why it was said that the stromal cells can differentiate into endocrine cells. Female sex hormones, we've already talked, is estrogen and progesterone. Right? With the increase in the age of the women, the collagen fibers in the cortical stroma gradually increase. So with age, the collagen fibers which were in between the stromal cells, they increase in number, and the stromal cells, they decrease in number. That is, fibrosis is something that does happen with age in most other organs as well. Right? And these are changes that are happening with age. The stromal cells decrease in number and the collagen fibers increase in number. By the end of the reproductive age, at menopause, the cortical stroma consists almost entirely of collagenous fibers, so there has been this high degree of replacement. So these were some major things we talked about. Now we will talk about the ovarian cycle briefly, and then we will come on to the follicle. This is important because when we draw it, we need to know how they look. The ovarian cycle, well, first of all, reproductive period of life begins at puberty and ends at menopause. And so the ovaries of every female will be undergoing these cyclical changes. And this is part of the ovarian cycle. It is a rhythmically recurring cycle, and it is 28 days long, just like the menstrual cycle, and it is divided into two phases, which you probably did already when doing the menstrual cycle: the follicular phase and the luteal phase. The follicular phase and the luteal phase. During this follicular phase, which is the first phase shown over here, right? And this is also known as the pre-ovulatory phase, because ovulation is at the 14th day of the 28-day cycle, and this is the time period before it. So this is given the name the pre-ovulatory phase, pre-ovulation, right? And many ovarian follicles, they undergo a growth process in both the ovaries. Normally this process results in the ovulation, the release of a single ovum from one of the two ovaries, and the two ovaries alternate with each other, meaning first month of the year, right ovary, then normally second month of the year, the left ovary, third month of the year, right ovary. So there's an alternation. Normally only in a month only one ovary ovulates, right? And the growth of the ovarian follicles occurs under the influence of the FSH, which is produced by the anterior pituitary. This is information that you're already familiar with. FSH is literally follicle stimulating hormone. So the growth of the follicles is because of FSH. And if memory serves you, when you're doing endocrinology and you will talk about all of the hormones produced by the pituitary, you will talk about TSH and you will talk about FSH and LH, which are the female gonadotrophic hormones. Now the growing follicles themselves, they're going to be producing certain hormones. They're the estrogens, which is why it is said that the ovaries are also endocrine. They also produce the female sex hormones, but they only do so at a particular stage of the ovarian cycle, in which the follicles are growing under the influence of FSH, and then they're able to produce estrogen. Which estrogen do they produce most? Estradiol, which is one form of estrogen, which is the most potent form. So this is something of an MCQ. MCQ. Estradiol, the most potent. Normal length of the follicular phase is 14 days. It is pre-ovulatory. Ovulation is at the 14th day of the cycle. Till 14th day, it is a pre-ovulatory / the follicular phase. Talking about the luteal phase, which is after the 14th day mark, after ovulation has occurred, the post-ovulatory phase. And now there is a formation of the corpus luteum, which is a temporary endocrine structure. Corpus luteum is formed from the remains of the ovulated follicle. After the ovum has been ovulated, the remains of the follicle basically become the corpus luteum. And the corpus luteum is going to be producing a lot of progesterone. In the initial parts of the cycle, there is a lot of estrogen being produced, and after the 14-day mark, there is a lot of progesterone being produced. Still lots of estrogen too, but progesterone is still the main actor. Length of the luteal phase is relatively constant at 14 days because if the ovum is not fertilized, the corpus luteum stops the secretion of progesterone after about 10 days of its formation and then rapidly degenerates. So stops functionality after 10 days of formation and then has a rapid degeneration, so approximately 14 days of the luteal phase. An additional almost 4 days for the degeneration to be completed. So that is something of an MCQ. How long after the formation does it begin to degenerate in the case of non-fertilization, when fertilization has not happened? Talking about the follicles now, because this is information that you will need when you're actually drawing the diagrams. So ovarian follicle is a spherical structure, right? Obviously, right? It contains an immature ovum, the eucyte, which is surrounded by epithelial cells which form a covering around it. And again, this is not information that is particularly new to you. In the growing follicles, a connective tissue covering is also present outer to the epithelial coat. Again, this is the theca. The ovaries of a female child contain only one type of ovarian follicle. So we're talking about the female child, one who is pre-puberty. Right? And such a child's ovaries will contain only the primordial follicles. So only primordial follicles, primordial follicles. However, during the reproductive period of life of an adult female, after puberty, 3 varieties of follicles can be seen in the ovaries. Each of these 3 types represents a different stage in follicular development, and all of them will be at different stages at any one time. So in one histological section, you might end up seeing all 3. So how many do we have? 3 types of follicles, and all 3 might be seen in a section of the ovary because they're representative of follicular development, and not all of the follicles are developing at the same rate. During the first days of each ovarian cycle, a number of primordial follicles, they begin to grow in both ovaries, and these follicles are titled growing follicles. Initial time of any ovarian cycle, some primordial follicles or several primordial follicles, they begin to grow, and now they're known as, what name is given to them? They're known as the growing follicles. And they're like, I guess they're 20 to 30, 15 to 20, my apologies, 15 to 20. Right? What happens next? Now, normally only one of these becomes mature, fully mature, and ovulates, approximately in the middle of each ovarian cycle at the 14th day. All other growing follicles in both ovaries undergo a process known as follicular atresia. They degenerate basically. Alright. As a result of this process, the follicular cells, they die by apoptosis. So in follicular atresia, there is apoptosis, programmed cell death. apoptosis. The ovum undergoes autolysis, self-digestion, and the zona pellucida collapses. We'll be talking about the zona pellucidaite, which is the glycoprotein layer, and when that is no longer needed because another one has become fully mature, that too degenerates, the ovum autolyzes, and all of the cells, the follicular cells that are surrounding the ovum, they undergo apoptosis. So the granulosa cells, follicular cells around the ovum, they apoptose. The ovum itself self-digests, and the zona pellucida outside of the follicular cell layer basically degenerates. This is follicular atresia summarized. The remnants of the degenerated follicles are engulfed and disposed of by the macrophages. So the macrophages have a cleanup role over here. They're going to eat up the remnants of the degenerated follicles. Alright. As already explained, the remains of the ovulated follicle, they do not degenerate. They become what is known as the corpus luteum, and this is a temporary endocrine glandular structure, something that can come up in an MCQ. Then we're talking about the actual follicles, starting with the primordial follicle, which was the only type of follicle present in the ovaries of a female child, one who was pre-puberty. A primordial follicle contains an immature ovum. A primordial follicle contains an immature ovum. An ovum that is not yet matured into the final stage, which is the oocyte. And it is actually a primary oocyte arrested in the prophase of meiosis 1, so it is not even completed meiosis 1. So to give the details over here, so it is a primary oocyte which is stuck in prophase of meiosis 1, and that is the status of the ovum, the immature ovum. Let me complete this information. So this is the functional status, and you must not forget this. Now this is something that can actually be asked in an MCQ about the primordial follicle. The oocyte of a primordial follicle, also it is surrounded by a single layer meaning just one layer of obviously follicular cells, but what kind of it? A single layer, and flattened follicular cells meaning squamous. So when you are drawing a primordial follicle, you kind of draw it like this. You represent the ovum, you draw in the nucleus, and then you represent the follicular cells as a single layer of flattened cells around it. And this over here is a representation of the primordial follicle as you would draw it. Now take a look at this. Now the oocyte of a primordial follicle measures 25 to 30 micrometers in diameter. So this is about 25 to 30 micrometers, 25 to 30 micrometers, right? And contains a large vesicular nucleus with a prominent nucleolus. It's hard to show that it is, you can see it is large, you cannot quite see that it is vesicular, unfortunately, because in this particular kind of way. And it is a prominent nucleolus, again, which is a bit difficult to show. And why is there a vesicular appearance? So the nucleus, it is due to finely dispersed chromatin of the primary oocyte arrested in meiosis 1, right? Because it is not in a condensed form as happens in the later stages of meiosis, so it's finely dispersed chromatin, and so it is pale staining. That is what vesicular really means. Cytoplasm of the oocyte. The cytoplasm of the oocyte contains abundant mitochondria. So there is a lot of mitochondria. which is going to be producing energy. So there's a lot of mitochondria, there is extensive RER, which is going to be responsible for protein synthesis of all things, and there are also a lot of lysosomes, my apologies, numerous meaning multiple, a lot of lysosomes. So we're having lots of mitochondria for energy, we're having lots of RER for protein synthesis, and we're having lots of lysosomes for degenerative purposes. Now the follicular cells which are surrounding the primordial follicles, which in this situation are single layered and also squamous, they're given another name, the granulosa cells. Sometimes people confuse it because they're not aware that these are the same things. And they're squamous epithelial cells that completely surround the oocyte and are attached to each other by desmosomes. Desmosomes basically are a type of cell junction, which are adhering type of junction. Now also the follicular cells happen to lie on a basal lamina which separates them from the stroma of the ovarian cortex. And this means that when we are drawing a primordial follicle, and we draw it as being spherical, we draw it as having a large nucleus, which is a vesicular nucleus, because the ovum happens to be stuck in meiosis 1 prophase, so there is finely dispersed chromatin. To show the follicular cell layer, you draw a single layer of squamous cells because it is simple squamous epithelium, right? So you draw it in this manner. Actually, because it is a bit hard to draw it in this manner if you're supposed to show the basal lamina, so you just have to show the fact that the cells are flat, and then you draw the basal lamina like this, which is a single layer that is covering it, and that is separating the follicle from the stroma. And as I've already told you, you divide the cortex into the stroma, and you divide it into the follicles, and the stroma will have the stromal cells, it will have the fibroblasts, the macrophages, lots of collagen, and lots of reticular cells. Now we're talking about growing follicles, which were the set number of primordial follicles that began development at the start of an ovarian cycle, and stimulation was because of FSH. Obviously, for anyone who has done embryology in first year, these are some obvious things. And this is secreted from the anterior pituitary gland of the female and starts at the age of puberty. And the follicular growth is characterized by alterations in the oocyte, so at which parameters does FSH have an effect. There are changes in the oocyte, and there are changes in the follicular cells because of the effect of FSH on the growing follicle. Alright. In addition, the stromal cells surrounding the ovarian follicle become modified. As previously mentioned, the stroma cells, which are spindle-shaped cells in the stroma of the ovarian cortex, they become modified to form the theca folliculi around the ovarian cortex. So the third change is that of the stromal cells to form the connective tissue capsule, the theca folliculi. Anyways, moving forward, as a primordial follicle grows, it is currently a growing follicle. Which developmental stages will it pass through? Unilaminar primary follicle, we'll talk about all of this. Unilaminar, lamina is a layer, uni means single. Unilaminar primary follicle, multilaminar primary follicle. So these are of the same stage, substages basically, because this is still primary follicle. Both are case of primary follicles. One is unilaminar and the other is the multilaminar stage. And there is a secondary follicle, and after that we'll consider some other things. Secondary follicle, this is the third stage. Now we talk about the unilaminar primary follicle. Unilaminar primary, that's it. Growth of the primordial follicle leads to changes in the oocyte and the changes in the follicular cell, something I've already said. Now the oocyte will enlarge in size. Previously it was stated that the size was 25 to 30 micrometers, and it will now attain a diameter of about 120 micrometers. Interesting, right? That the nucleus becomes bigger. It was already stated to be quite big. The mitochondria, which were already plentiful, they increase in number. The RER, which was already extensive, becomes even more so. And the Golgi complexes similarly increase in number. And there is another change. They migrate to the periphery of the cell. Special secretory granules called the cortical granules, they appear in the most peripheral part of the cytoplasm. This is interesting. Appearance of the cortical granules in the peripheral part of the cytoplasm. P for periphery. And this will make sense in the actual ovulation, in the actual fertilization, because these cortical granules will then have a role, and they appear in unilaminar primary follicle stage. And the unilaminar primary follicle stage is a subgrouping of the growing follicle, because growing follicle is a collective term, follicles undergoing development. During the process of fertilization, the cortical granules are also released by the oocyte into the Zona Pellucidaite to prevent polyspermy. They ensure that only a single sperm will be responsible for fertilization, because you don't want multiple sperms fertilizing the same egg. That would lead to a lot of bad stuff. The follicular cells increase in number by mitotic division and also change their shape from squamous to cuboidal. So what happens in this stage? There is an increase in number of the in terms of the follicular cells. They increase in number, right? In terms of their shape, they go from, this was number one. Second point, they go from squamous to cuboidal. So they go from squamous to cuboidal, which is interesting because cuboidal is also the surface epithelium of the ovary itself. So these are the two things we considered during the unilaminar primary follicle stage. Right. Thus, a unilaminar primary follicle is surrounded by a single layer of cuboidal epithelial cells. It's not stratified yet because the name suggests unilaminar. There's still proliferation, but the size of the oocyte itself has increased, so you do need more to fully cover it. And the cells are now cuboidal. So when you draw the unilaminar primary follicle, it's something like this. I'm drawing this a bit bigger to kind of drive the point home that the size of the oocyte has increased from 25 to 30 micrometers to 120 micrometers. And similarly, the size of the nucleus has increased. The cytoplasmic content, the mitochondria, the RER, and the lysosomes have become more extensive. And there also has been a change not only in the oocyte but also in the follicle cells. They have proliferated and they've changed their shape too. My apologies. They've changed their shape too. This is actually not quite true. Now this is fine. What makes a cuboidal cell a cuboidal cell? When its dimensions are identical in different planes. So this is incorrect, these are correct. So this is the representation of it completely covers it, and this will then be a representation of a unilaminar primary follicle. So a primary follicle is going to be characterized by cuboidal epithelium. In unilaminar stage it will be a single layer. In multilaminar it would be stratified. Primordial will simply have squamous epithelium. Alright, I'm hoping this kind of makes sense. Now, obviously, we must not forget the basement membrane. The granulosa cells, aka the follicular cells, are joined to each other by the gap junctions. Previously, we stated that they were joined to each other by desmosomes to ensure that they were adherent. Now, there is gap junction, meaning there is free communication between the follicular cells. Gap junctions between the follicular cells, meaning that there is a measure of free communication. Now, a layer of amorphous homogeneous material appears between the oocyte and the granulosa cells, and this layer is known as the Zona Pellucida, and it is 5 to 10 micrometers thick. So the name of it, Zona Pellucidaite, is an amorphous layer. It is not cellular, and it is homogeneous, meaning the staining is the same all around. And it is in, where is it? It is between the oocyte and the follicular cells. It's not over the follicular cells. I might have misspoken initially. Right? So the thickness is 5 to 10 micrometers. So it's pretty thick, considering that 10 nanometers is the cell is the membrane diameter. It takes a deeply acidophilic stain. Acidophilic means pink, for those of you who are not familiar with this kind of stain. This color, pink color stain. Right? The Zona Pellucida contains 4 glycoproteins because it is in fact a glycoprotein layer. ZP represents Zona Pellucida 1, Zona Pellucida 2, Zona Pellucida 3, Zona Pellucida 4. So this is convenient. And they are secreted by the oocyte. The Zona Pellucida layer is itself produced by the oocyte in order to cover it. And so these proteins are being produced by the oocyte, which does make sense why it needs the RER and why it needs the mitochondria. During the process of fertilization, some of these proteins will serve as receptors that bind to the proteins present on the surface of the sperm. So there will also be a measure of receptor function, which is a possibility. Thin tapering processes known as philopodia from the follicular cells and the microvilli, they penetrate the Zona Pellucida and communicate with each other by gap junctions. So something of an MCQ, philopodia from the follicular cells, they penetrate into the Zona Pellucida. And from the other end there is the... So now talking about the multilaminar primary follicles. Multilaminar primary follicles. As growth of the ovarian follicle continues, the follicular cells, they proliferate further and they give rise to several layers. Now this is obvious. So now it is known as multilaminar, and the multiple layers are given the name stratum granulosum, or the layer of granulosa cells. Stratum means layer. Stratum granulosum, referring to the stratified layer of granulosa cells. As the follicular growth progresses further, the stroma of the ovarian cortex, the cortex was divided into the stroma and the follicle. So now we're considering the stroma, because in this stage we have the formation of the theca folliculi after the stratification has been achieved. The stroma of the cortex begins to form a sheath around the ovarian follicle. This is given the name the theca folliculi. Stratum granulosum is separated from the theca folliculi by a well-defined basement membrane. So there is a basement membrane in between. Theca folliculi itself will differentiate into an inner layer, the theca interna, and an outer layer, the theca externa. So I'm hoping you're keeping up. Talking about the theca interna, consists mainly of roughly, so its approximate shape, roughly cuboidal cells, which are secretory cells. Secretory cells, and they're known as the theca cells, interestingly. Cuboidal is the shape, secretory is their functional capacity. Right? In addition, the theca interna, because it is a connective tissue capsule, will have fibroblast. and you represent fibroblasts mainly with dots in histological diagrams because you can only see their nuclei. There's also a lot of fibroblast in the theca interna, a lot of collagen because it is still connective tissue, and there's also a rich network of fenestrated blood capillaries. This is very significant that there is a rich network of fenestrated blood capillaries in the theca interna. So talking about the theca interna, that is the internal layer of the theca folliculi. The theca folliculi is made by the modification of the stroma. Theca interna has a lot of stuff. It has the theca cells, right, and that is simply the cuboidal cells, which are secretory cells, right. In addition, there's a lot of collagen, there's a lot of fibroblast, and in addition to the theca cells, the collagen, the fibroblast, there is a network of fenestrated capillaries because this is a well vascularized layer. So basically there is a good blood supply. Now the theca cells, we're talking about the theca cells which have the endocrine function actually, which we mentioned at the start, and which are roughly cuboidal, and they're the transformed stromal cells of the ovarian cortex. They exhibit characteristics of steroid producing cells just as they should because they're in fact steroid producing cells. And what would those characteristics happen to be? Let's talk about those. Numerous lipid droplets because that is the raw material, the reactant, the precursor. Droplets because lipid is stored in the form of droplets because the environment is aqueous and droplets are hydrophobic, lipid droplets, so they are in the form of droplets. That is one characteristic of steroidogenic cells possessed by the theca cells of the theca interna. Additionally, there is a lot of mitochondria with tubular cristae. Mitochondria because energy is needed for the steroidogenic function. A lot of tubular cristae because there are specializations within mitochondria that need to produce a lot of energy, which include increasing the surface area to suit the needs via increasing the tubular cristae. And then the third point is there's abundant SCR, not RER because that is for the protein based hormones. These are the steroidogenic hormones, so there needs to be extensive SCR. So there's a lot of lipid droplets, a lot of mitochondria with their tubular cristae, and also a lot of SCR, which is just what you would expect out of a steroidogenic cell. Now, what do they synthesize? Androstenedione.